The world's largest medical research lab isn't where you'd expect. At all times and places, from ocean depths to mountaintops, in air, on land and underground, chemical experiments have occurred through competition, cooperation or symbiosis between species for billions of years. These experiments sometimes result in the creation of toxins, chemical weapons, which for the plants, bacteria or fungi that develop them are formidable means of defense against predators. These are their shells, their claws, their shields, their fangs. Because unlike most animals, which can run, crawl or jump away in the face of imminent danger, for them there's no way to escape, to fly away or to bury themselves. To protect themselves, evolution has created a range of fearsome chemical weapons through random molecular combinations. Just consider this. Some bacteria living in the soil produce ivermectin, a molecule with powerful insecticidal properties, and it's a nightmare for the central nervous systems of most nematodes. Others can protect themselves from fungi by producing molecules capable of literally liquefying their cells. By the way, if you are infected by a fungus, then there is a good chance that you will be prescribed bacteria with the gentle name Streptomyces nodosus. Some fungi produce compounds that break down bacterial cell barriers. Remarkably, these chemical weapons can be deadly for some, but harmless or even helpful for others. So, if you suffer from a bacterial infection, you might be treated with toxins synthesized by certain species of mold. That's penicillin. Treatments for eczema have been found in bacteria, and treatments for cholesterol in fungi. A substance can have very different effects depending on the species that consumes it. Wild boars, for example, can gorge themselves on acorns, while the tannin they contain is a powerful poison for humans and ruminants. Everyone knows the cup of igi, which is used as a symbol of pharmacy in many countries. But what's funny is that pharmakon, a word of Greek origin, means both to prepare medicines, magic potions, but also to poison and to bewitch. In fact, even though the vast majority of medicines were discovered only very recently, a small part comes to us from the dawn of time. We know that more than 30,000 years ago, Neanderthals were already using some medicinal plants. Since antiquity, pharmacopoeias have been developed all over the world. Care was provided by priest-doctors, healers, and shamans. By trial and error, they discovered, tasted, tested, and maybe even poisoned themselves a little. And knowledge was passed down over time orally, from generation to generation, and then in writing. To realize the incredible journey behind medicines, which may seem harmless to us, let's take the example of aspirin, which partly comes from the bark of the white willow. 3,500 years ago, Egyptians used this plant to relieve pain and fevers, but it took over 3 millennia before its properties were studied. And it was only in the 19th century, after finding a nearly equivalent substance, salicylic acid, in meadowsweet, that people began to synthesize aspirin. In the end, it would take many more years of research to extract the true active ingredient from willow, stabilize it, and be able to produce aspirin in large quantities. And it was only more than a century later, in 1971, that people finally understood how it works. To understand the progress made, consider that in 1900, the average life expectancy in France was 45 years. By 2017, it had risen to over 80 years. And who do we largely have to thank for all this? Doctors, vaccines, researchers, yes. But also to plants, fungi, and bacteria. Because 70% of the medicines we commonly use contain chemical compounds that originally come from these organisms. Strangely, even though there is a greater variety of animal species on Earth than there are species of plants, bacteria, or fungi combined, almost all of our remedies come from the latter. How did we discover all these molecules hidden in nature? Well, most of the time it's thanks to what we call serendipity. That is, a happy accident that allows a prepared mind to discover important things they weren't specifically looking for. That's how, while trying to treat angina in the early 90s, we discovered how to induce prolonged erections. Because as strange as it may seem, the discovery of Viagra was accidental. It came from the surprising side effects of a drug that was originally supposed to treat angina. The funniest part of all this is that it was recently discovered that what became Viagra has the side effect of relieving jet lag in hamsters. Expect to find something other than what you're looking to use. It's very often by digging into this library of molecular diversity created by nature that we discover drugs, deadly poisons, and potential medicines. Because the line between these three is often thin. Here is the jararaca, a fearsome viper from Brazil. Its venom is so powerful that a bite from it can lead you to certain death. Yet, from studying it, a molecule was discovered that can fight against high blood pressure. A molecule that has probably saved more lives since its discovery in the 1970s than the total number of deaths caused by viper bites throughout the entire history of humanity. The deadly poisons of yesterday can become, for some, the miracle drugs of tomorrow. Henbane is a plant that can be lethal, yet, when precisely dosed, it can be used as a treatment for coughs. Take something like aconite. Actually, no, definitely don't take something like aconite, because this plant contains particularly dangerous alkaloids to the point where just a few grams of its roots would knock out any human. And yet, paradoxically, extracts from this plant can be used as an antidote to scorpion venom. Belladonna is also extremely toxic. Yet, ophthalmologists use it to dilate the pupil. The problem is that sometimes, even if we find effective treatments in plants, very often the molecules we're interested in are present in infinitesimal amounts. This can cause major problems. The yew is a tree that can live up to 1,500 years and whose leaves and bark have been known to be toxic since ancient times. In the 1970s, it was discovered that it contains a toxin, Taxol, which is very effective in the fight against certain cancers. The problem is that the amount of this molecule in the tree is very low, on the order of a few grams per ton of dried leaves. So, in the 1970s, to produce the precious molecule, to obtain just 2 tiny kilos of Taxol, it was necessary to cut down 12,000 trees. Fortunately, we eventually managed to synthesize this molecule in the laboratory, which saved the unfortunate plant from being cut down and made it possible to create derivatives, new medicines that are even more powerful and effective. Today, the problem is the same with the Madagascar periwinkle, which contains a molecule called vincristine, which is also very effective in the fight against many cancers. The fact that this molecule exists in nature is a blessing. But you have to process a kilo of dried periwinkle leaves to get 10 milligrams of vincristine. The molecule's structure is so complex that the CNRS didn't reproduce its active ingredient in the lab until the late 80s. The active ingredient is the part of the medicine that actually has therapeutic effects. These substances are generally present in very small amounts compared to the rest, which is called the excipient, meaning what gives texture and taste, while avoiding any interaction with the active ingredient. For example, the caramel-like viscosity in a cough syrup. Today, from discovering a therapeutic molecule to developing a drug and releasing it for a specific disease, the process is lengthy and challenging. To sum up, the first thing you need to do if you want to create a drug is to be persistent and, above all, patient. Because on average, the process between the discovery of the active molecule and the commercialization of a drug takes 15 to 20 years. Because first, you'll need to discover or synthesize the active molecule, find the right form, the right arrangement of atoms that will go to the right place to fight the disease you want to target. It's a bit like a Lego game where you have to be creative, finding the right keys for the right locks by testing thousands of molecules before discovering one with pharmaceutical potential. On average, 2 to 4 years of research and development are needed to discover new plants in the field, analyze their effects, or simply create and synthesize entirely new molecules in the laboratory. After that, you have to file patents, which are intellectual property rights that last about 20 years. Then comes the clinical development phase, which lasts 6 to 8 years, during which products are tested. First on cell cultures, then on animals with genetic backgrounds similar to humans. Mice, rats, pigs, and monkeys. Next, it's the turn of healthy human volunteers to test the candidate drug in order to determine its toxicity, side effects, optimal dosage, and potential risks. And it often happens that drugs end up being sought after more for one of their side effects than for their primary effect. That's the case with minoxidil, a highly toxic substance used in very low doses as a treatment for high blood pressure. Roughly 10 years after this molecule was discovered, clinical trials showed some users experienced increased body hair growth. So laboratories began to use the properties of this product to treat hair loss. In short, after this step, the effectiveness of the product will be tested on a small number of patients. Some of the patients will take the real medication while the others will receive a placebo. This phase will determine the optimal dose of the medication and assess the benefit-risk ratio. If everything goes well, marketing authorization, or MA, will follow. It's the legal approval to market medications, but that's not the end. The active ingredient, therapeutic molecules, and excipients still need to be produced. What form should the medication take? Should it be made into capsules, a syrup, ampules, powder, sprays, or injections? Is it taken orally or inhaled? Which option is most effective and most acceptable? Once this step is finished, you might think that all that's left is to market the product and that's it. Except that you have to monitor, evaluate, and stay vigilant about potential side effects that might have been missed. That's pharmacovigilance. Because unlike the clinical development phase, where only a few dozen, or at most a few hundred people take the new product, once it's on the market, tens of thousands of people end up coming into contact with it. To understand how far we've come at this stage, you should know that out of 10,000 substances studied and tested in the lab, only 10 make it to the clinical trial phase. And only one ultimately meets all the criteria. To be marketed as a drug, how many unknown molecules, how many remedies invented by nature are still left to discover? Well, a lot. A tablespoon of forest soil can contain up to 10,000 different species of plants, microscopic worms, fungi, or bacteria, and most of these species have never been studied. Of the roughly 4,000 diseases whose molecular basis is known, just over 250 currently have treatments, and it is estimated that only 10% of organic compounds in the plant kingdom are known. Which is fortunate, because it means there are still plenty of hidden treasures to find, observe, and study, little gems of complexity that could one day save our lives. All that's left is to dig. Maybe to find something other than what we were looking for. Creating videos takes a lot of time. If you enjoy our work and want to help us continue, you can support us on Typee. Maybe one day we'll make a living from it.